Okay, thank you. Good afternoon. My name is Günther Wagner. I'm hosting the Ph.D. lecture series on the developmental evolution of reproduction. I'm particularly pleased today to welcome our speaker, Antonis Rokas, who is the Cornelius Vanderbilt Chair of Biological Sciences, of course, from Vanderbilt University, and he is going to talk to us today about the evolutionary regulation of gestational length. Antonis.
Thank you so much. Let me make the window so that doesn't block my screen. So it's thank you so much, Günther. It's a great pleasure to be here and to be talking to you about our research. And so, yes, so I'm an evolutionary biologist, and I thought I would introduce very, very briefly the lab so that you understand the perspective through which we approach the questions of the evolution of gestational length and other reproductive traits. And so we are interested in evolutionary genomics. And so we study the DNA sequences, the genome sequences of several different organisms, including mammals. And we're particularly interested in understanding the patterns and processes of evolution. And we use a combination of computational and experimental approaches. Experimental in my lab is typically high throughput omics types of approaches. And then with collaborators, we engage in also testing mechanistic hypotheses and so on. And my lab has sort of very broad interests. We're interested in phylogenomics. This is part of my background and training. We have projects on several different groups of fungi, and then we of course interested in the evolution of mammalian pregnancy. I'm also, you may have noticed, Vanderbilt University is in Nashville, Tennessee, and being an evolutionary biologist in the south of the U.S. comes with particular we find ourselves in a particular setting. Vanderbilt is about 2.5 hours away from Dayton, Tennessee, where almost 100 years ago, in 1925, a science teacher named John T. Scopes was convicted for teaching science in school kids. And some of the issues that sort of originated in this trial and the relationship between teaching of science in high schools and religion sort of trace back to that trial, trace back to the debates that started in 1925. And so together with several different colleagues, I'm involved in establishing a center for the study of evolution at Vanderbilt. And if you're interested, I can tell you more. There is a website, and our logo is inspired by the phylogenetic tree in the textbook that John T. Scopes taught evolution apparently from. And so that's sort of a very brief, a very short introduction on some of our interest and general approach to science. I'm here to tell you about evolution's influence on gestational length in mammals. And the basic question, the basic problem that we're trying to study can be summarized by this figure. And so on the x-axis, you can track the gestational age at birth. These are data from humans. And then the two different y-axes indicates the rate of neonatal deaths and the total number of live births in the human population. And so you can see that, around 39, 40 weeks of completed gestation, pregnancies maximize the number of births and minimize the number of deaths. And as soon as you move outside of this gray zone, so to speak, you start increasing the number of neonatal deaths. And we're not so particularly interested, especially in developed nations, about post full-term deliveries. We have C-sections, we have mechanisms by which we can induce pregnancies. But the big issue comes with births before 37, before 36 completed weeks of gestation. And the reason that that's a big concern is because we identify any birth in humans before 37 completed weeks of gestation as preterm birth. It's simply the single largest cause of infant death worldwide. And we really don't know much about it. For about 50% of the cases, we know the factors that contribute to it. For the rest 50, it seems like the cause is unknown. And we'd lack any interventions. There are really no known strategies that seem to work, and also the trait, preterm birth, the rates of preterm birth show huge, especially in the US, huge disparities along different ethnic groups, with African-American individuals tending to show preterm birth rates almost double those of whites or Asians or people from other ethnic backgrounds. Okay. Now, what we do know is that there are many factors that contribute to preterm birth in the study of humans. There are factors such as stress or infections, bacterial or viral infections, immune status of the mom, inflammation, and so on. But we also know that genetics plays a role. We know that from twin studies and from other genetic epidemiological studies. And that's the fraction, that's the part of the problem we're studying. That's not to say that all the other factors do not play a role. They're very, very important, and we know they contribute, but we're particularly interested in the genetic side of preterm birth. Now, when we consider, I think that's true for reproductive traits in general, but it definitely applies to gestation length. It's important to consider the differences of studying a reproductive trait relative to studying any other complex genetic trait. And so in contrast to, if you think about, say, cardiovascular disease, you have a particular tissue that is involved in the disease. You have a particular genome of the individual that has the disease that may have variants that contribute to disease risk and so on. In contrast, for when we consider gestation length, we don't know the tissue that's responsible. It might be the myometrium, it might be signals from the placenta, it might be signals from the baby, it might be signals from the mom. And so there are multiple tissues involved and two individuals being involved, both the mom and the baby. And then we have alleles, transmission of alleles. Some of the alleles of the mom are transmitted in the fetus. Half of the alleles of the dad are transmitted in the fetus, but the mom can also exert influence through the untransmitted alleles, right? Because she's carrying the pregnancy. And so depending on the allele we're talking about, so whether we're talking about the maternal untransmitted allele or the maternal transmitted allele or the dad's transmitted allele and so on, the effects, the influence on the pregnancy phenotype, such as gestation length, is different. So an untransmitted maternal allele can have an influence on gestation length through the mom, but not through the fetus. The transmitted allele could have effects through the mom and the fetus, and so on and so forth. And so it's definitely more complicated. In addition to that, if we consider the evolution of reproductive traits and the evolution of gestation length, it's important to acknowledge that pregnancy in humans is associated with two key evolutionary transitions, one of which is brain enlargement. There was obviously very clear brain enlargement in the history of primates, but also in the history of the human lineage. And then specifically in the history of the human lineage, there is the transition from a quadrupedal mode of walking to a bipedal mode of walking. And both of these key evolutionary transitions, with obviously major consequences, but for our species, have influenced, and to some extent, we don't know directly or indirectly, but have definitely left their footprints or their signatures on parturition. And so if we take a slice through the pelvis or different sections of the pelvis of different primates, ancient and modern. So this is modern chimpanzees. This is Australopithecus afarensis, Lucy, and this is modern Homo sapiens. You can see the differences in the planar views for each one of these sections. And so the baby, as it's born in chimpanzees, has no trouble escaping. There is no particular pressure, whereas in humans, the space is very tight. Okay, now evolutionary change is not only something that we can see in the fossil record or as we study the evolution of humans relative to those of other species, but it's actually ongoing. We see differences in terms of average gestational age, optimal gestational age at birth across different human populations. And so Asians tend to have an average gestation length of 39 weeks. White Europeans tend to have an average gestation length of 40 weeks, and that doesn't seem to be associated with any health disparities. And so we do see differences among human populations. And then we also see differences as we study a particular human population across time. So this is some classic work by investigators in different Italian villages where they had rates of stillbirth over time for the same villages. And what these plots show is, what the data look like, what the rates look like relative to birth weight, fetal birth weight across time. And so if you zoom in, and this is what this inset is showing here, is the zoomed in version of the bigger graph, what you can see is that in the 1950s, the shape around the optimal birth weight is pretty tight, right? So you go smaller birth weights or larger birth weights, and you very quickly increase stillbirth rate. But then as you go towards the 1980s and the 1990s, then the shape of the curve has flattened. And basically what we're seeing is, right, the advent of modern medicine. We can take care of some of these situations and reduce stillbirth rate relative to that. But of course, potentially this has evolutionary consequences in terms of changes in allele frequencies and the like. Okay, so to summarize this brief introduction I'm giving you about gestational length and its evolution, we know that complex genetic traits and diseases associated with them typically involve numerous genetic and environmental factors. That's well established. But in addition, when we consider pregnancy-associated traits like gestational length and pathologies like preterm birth, we need to take into account further complications, such as that we have 2 individuals and 3 genomes to deal with. We have multiple tissues that are involved that may be extensively communicating with each other. And then we have that in the background of very rapid evolution for major traits, specifically in the human lineage. And so I want you to have this in the background. You'll see some of these features coming up really quickly for some of the research. And so what I want to spend my time, the remaining 30 or so minutes that I have, is to talk about 3 vignettes of the work that we've been involved in as part of a larger group, in particular the March of Dimes Ohio Collaborative for the Study of Preterm Birth. So the first one is going to be on the genetic architecture of gestational length in European populations, and that's mostly the work of collaborators. We have very little involvement, but it really sets up nicely some of the questions that we have led, such as studying the effects of selection on human loci involved in the control of gestational length in humans. And then broader in the third vignette, broaden the perspective and talk about the evolution of gestational length in other mammalian species and across mammals in general. And so for identifying the loci, when we started the work, when our group, the Ohio Collaborative, started working on the topic, it was not much known about the genomic regions in the human genome that are associated with variation in gestational length that we see across humans. And so Gezang, Bo Jakobsson, and Lou Muglia and collaborators started a really large-scale study and conducted the first genome-wide association of gestational length as well as of preterm birth. Here I'm showing you the data, I think, for gestational length. And what they found by studying a population of 44,000 women or so, they found significant evidence for association in 6 loci associated with preterm birth. Now, again, as I mentioned, there are maternal contributors, there are fetal contributors, and so on. And so we can use, and Gezang has really innovated on this, to use Mendelian randomization and other statistical genetic techniques to basically understand pregnancy outcome and take into account confounders that may be influencing maternal phenotypes and pregnancy outcomes, to identify direct causal effects of maternal phenotypes on pregnancy outcomes, and to also identify and distinguish direct fetal genetics effects on pregnancy outcomes. And so we can use, or can use, Mendelian randomization to disentangle effects of non-transmitted and transmitted maternal alleles on the mom, on the baby, as well as transmitted maternal alleles on the mom and on the baby. And so With that in mind, with this toolkit at hand and with an expanded cohort, not only of women, so now we've quadrupled the size. We're now almost to 200,000 women and about 140,000 moms and babies. Now the number of variants that we have associated has quadrupled too. So instead of 6 loci, we now have 22, and instead of a few variants, we now have 24. But now we can start separating, having the parent offspring data. We can start separating and do GWASs on just the maternal loci, just on the fetal loci, and identify contributions of the different loci and whether they act only through the maternal genome, only through both maternal and fetal genomes, or only through the fetal genome. And it's important to emphasize that the variants and the loci that are being associated confirm the previous discoveries and, of course, expand them. And when we look for, what these variants do and what particular tissues they're enriched in their expression, what we find is that reproductive tissues are highly enriched. So we see uterine tissue, we see cervix, ovaries, endometrium, and so on, the tissues that we really expect to be enriched in loci associated with gestational length and preterm birth. So that's good news in that we keep expanding the number of loci that we're finding. It also indicates that, right, as we increase the size, we're going to find even more loci and explain the bigger portion of the variation that's observed in gestational length. Now, one interesting thing that has been found is that we can use SNPs, right, for any variant that has been associated with gestational length. We can look at its effects on gestational duration as well as other traits. And one particularly interesting trait is birth weight, how the weight of the baby. And so we can take maternal non-transmitted alleles, and we've done the analysis with transmitted alleles as well, and measure the effects of the maternal effect on gestational duration. And then we can also take, for the same SNPs, we can measure the maternal only effect on fetal birth weight. And we see a nice positive correlation. But what's interesting is that if we take the same SNPs, right, so the x-axis hasn't changed, the effect of maternal non-transmitted alleles on gestational duration, that's the same data. But for those SNPs, we calculate the effect that acts through the fetus on birth weight, then we see an inverse relationship. Okay, so alleles that increase gestational duration tend to decrease birth weight in the fetus through acting through the fetus. And this is really evidence for antagonistic pleiotropy, right? So the same variant may contribute to the increase of one trait, but contribute to the decrease of another trait. Such evidence or antagonistic pleiotropy has been for a long time thought to be associated with particular human diseases and trade-offs, right? That we can't optimize every trait possible. Typically, we've seen antagonistic pleiotropy in diseases associated with neural system development and nervous system development, such as, a classic case is this gene that's human-specific. It has originated from a human-specific duplication of an existing gene. It shows strong signatures of expression and has been functionally associated with the expansion of neocortex. But the duplication that has been created make this region susceptible to microdeletions, and these microdeletions tend to be associated with disorders, disorders of the nervous system. And so something that's potentially adaptive has also created the potential for disease, and that's illustrated as antagonistic pleiotropy. And so it's exciting that we're seeing the same with gestational length. Okay, so now that we have, or that we're beginning to identify genomic regions associated with variation in gestational length, we can start asking evolutionary questions, such as, what are the evolutionary forces that have shaped the variation in these regions, okay? And so we can take the variants associated with gestational length, and we can apply several different evolutionary tools that aim to detect signatures based on patterns of sequence variation in human genomes, try to identify signatures of evolutionary processes in human genomic data. And so that's something that we have been working on quite a bit in collaboration with Tony Capra's lab in the last few years. And so just to briefly summarize some of what these techniques do, or to give you a flavor of what they do, imagine you have a set of alleles that you sampled from a human population. You're going to see variation between these alleles. Some of the variants, some of the single nucleotide polymorphisms are going to be neutral, right? So they're not be either beneficial or detrimental to the fitness of this organism, or to the carriers of these alleles. But occasionally you have a mutation that's beneficial to the carrier. And so if that's favored by selection, and if certain conditions hold, what we expect to see over time is that the population will start having multiple individuals, right? So the frequency of the allele will increase in the population. And so as we sample the population at a later point in time, we're going to see an increase in the frequency of that allele. Now, because that beneficial mutation, when it originally occurred, it was linked. It happened to occur in the neighborhood where there were these two other neutral variants. These other two neutral variants, because of the infrequency of recombination, will start hitchhiking along with that beneficial mutation. And so over time, we expect, if the beneficial mutation gets fixed in the population, we expect to see a pattern where around the beneficial mutation, there is a dearth, there is a lack of variation, because not only that beneficial mutation, but also the variants that were linked to it have been fixed in the population. Now, of course, recombination will occur sometimes and create some variation in these patterns, but that's one of the types of patterns that we look in human population data. We look for regions of the genome that may be depleted in variation relative to what we would expect for a region of particular characteristics. And we have different tools that look for different patterns in the human sequence data and try to identify or tie them to particular evolutionary processes. So here is a table. I don't expect you to memorize any of this or to look at it in great detail. But what I want to point out is that the patterns that we can detect reflect potentially the action of several different evolutionary forces, and then enable us to look back at different time scales. And so some tests seem to be really good at detecting recent events of positive selection. Others seem to be really good at detecting more ancient events. And then, of course, we have several different evolutionary forces that we can infer the action of. Now, before I go any further, I need to emphasize two caveats associated with these types of analysis. The first is that I think it's obvious to you that we're studying patterns of variations and we're trying to infer processes. And so there is always, there are potentially many different processes that can generate the patterns that we observe. And so that's something that we always have to keep in mind. Evolution is not always clean or easy to infer. And then the second is that we're studying selection, and we're trying to see the evidence for selection on genomic regions associated with preterm birth. But it's important to remember that genomic regions are involved not only in controlling variation in gestation length, but potentially many, many other characteristics, right? So in other words, they are pleiotropic. And so we don't know if we infer, say, evidence of positive selection or a particular genomic region that we've identified through a genome-wide association study on preterm birth or on gestation length. We don't know whether selection was specifically to reduce or increase gestation length. Okay, all we know is, all we can infer is that selection acted on that particular region, and there needs to be additional work to potentially validate whether selection was for preterm birth. And so having made these two caveats, I can ask the question of, right, what are the evolutionary forces then that have acted on gestation length associated loci? So we can take the work of Ge and Paul and others and apply our metrics and develop pipelines that enable us to measure the evolutionary forces that have operated on that. And so very briefly, what the approach that Abi Labella, a former postdoc in the group, Eben Abraham, former grad student, Tony Capra's lab, did, working together, was to start, and for every region, they started focusing on spontaneous preterm birth. They would take the particular region, and they would generate, by sampling genomic data from the 1,000 Human Genomes Project, by sampling 5,000 other regions that had similar characteristics in terms of minor allele frequency and linkage disequilibrium. The reason for doing that is that we've seen that not adjusting for those creates biases in the inferences of selection. And so first we create a population of regions, if you like, or a sample of regions, control regions, that we can do the same type of analysis as we'll do in our region of interest, and then evaluate signatures of selection and whether they're statistically significant. And so we identify these 5,000 control regions. For each one of these regions, including the original one, we can measure selection, and we can generate distribution, and we can see whether our particular region's score in terms of measuring the selective force is significantly different from the one of the control regions that have been adjusted for allele frequency and linkage disequilibrium. And we can do that not just for one region that has been associated with preterm birth, but for all the different regions that have been associated with preterm birth. And then we can do all kinds of functional annotation for regions that appear to be significantly influenced by a particular evolutionary force. And when we do this, we get a picture that looks like this. And so each column corresponds to a different genomic region that shows, through genome-wide association studies, evidence of association, significant association with preterm birth. And then each row is one of the different metrics that we use to infer the evidence of positive selection or balancing selection or local adaptation or negative selection and so on. And whenever you see these stars, the different colors in the cells indicate enrichment or depletion of the presence of different evolutionary forces relative to the background regions. And what I want you to see is that, you know, the evidence that we have is that different regions show different types of selection. So we have some regions that show evidence of positive selection. We have some regions that show evidence of balancing selection, others that show evidence of local adaptation. Of course, we have lots of regions that show evidence of purifying or negative selection, and so on. So in other words, we're seeing a mosaic of evolutionary pressures. There isn't a single signature of selection operating on loci associated with gestational length. We see all kinds of evolutionary forces, all kinds of modes of selection operating. Just to give you one example of one such region that shows evidence of selection, this is a gene called OPRL1, opioid-related nociception receptor 1. And we know from previous studies that it has been shown to be involved in myometrial contractions during delivery. There are two alleles. The T allele is protective, and the C is the risk allele. The protective allele is unique in humans, so you only find it in the human population. You don't find it in the genomes of closely related primates that include several different individuals. You can see a signature of selection in the African population for the protective allele. And that's just one example. When we go through the data, we find several different examples that reflect the different evolutionary processes. Okay, and so the take-home message that I want you to remember is that, at least for gestational length, the regions that are associated with that trait tend to harbor really diverse evolutionary signatures. And we can perhaps use these evolutionary hypotheses to develop hypotheses about how these regions may contribute to preterm birth risk. Okay. Now, the next step that Abin and Abi did was to say, well, okay, we now have a way to identify the action of different modes of selection, or to predict whether a particular mode of selection has acted on a particular genomic region associated with a complex trait. But can we apply a similar type of approach on an entire trait? So in other words, if we apply an evolutionary measure on a particular GWAS that's been associated with a trait, say gestational length, can we compare it, or can we gain an idea of how much selection has shaped, or a particular mode of selection has shaped that entire set of genomic regions that are associated with that trait. And so we can do a similar type of statistic where we generate regions that look similar in terms of minor allele frequency and linkage disequilibrium to the regions that have been identified in the original GWAS, generate the set of control regions, generate summary statistics, and evaluate significance based on the empirical data that we've analyzed. And the power of this is that we can do it not just for gestational length, but for the 900,000 GWASs that have been done on all kinds of different traits. And that's what Abin and Abby have done, is they can go and start analyzing several different GWASs, and they've done it in this preprint that was released late last year. And so we can start asking questions such as, are reproductive traits showing similar evolutionary signatures as the mosaic influence of different modes of selection as preterm birth? And the answer is yes. But what's really interesting is when you start comparing particular, say reproductive traits with other types of traits, such as traits associated with hair and color. So there we see very strong signatures of negative selection, very strong signatures of local adaptation. Perhaps this is not surprising for traits that we know vary with respect to environment and latitude and longitude, okay? But we can also go to, say, diseases that are late onset, such as there are several different GWASs for late onset Alzheimer's, and we can look at genomic regions associated with late onset Alzheimer's. And they are, as perhaps you might expect for a late onset trait where selection might be expected to be weak or non-existent, we don't see any signatures of selection. And so we believe this approach enables us to compare, to look for signatures of selection or reproductive traits, but also compare and test this hypothesis about whether reproductive traits are unusual in the evolutionary forces that operate on them relative to other traits. And you can see I slipped into thinking or speaking about selection on traits rather than selection on genomic regions associated with these traits. I think it's really selection on the genomic regions that were detected. Okay, so for the last few minutes, I want to sort of broaden the scope and move away from the human species and start talking about other mammalian species and what do we know about the evolution of gestational length in other organisms. And so first of all, when we started the project and our involvement with the March of Dice Ohio Collaborative, a lot of the medical folks that we would talk to had this notion that preterm birth and this variation in gestational length would be something that would be really human specific, that this disease of preterm birth doesn't exist, or the syndrome doesn't exist in other species. And in some ways they're right. We don't see often the severe impact that we see for human preterm birth in other organisms. But in terms of studying the traits and its variation around the optimal gestational length for the different species, these are data that we compiled a while back from several different species that we could identify population data. And these box and whiskers plots show one standard variation from the mean and then two standard variations from the mean. And so in humans, and they're normalized so that we can compare the gestational length and the variation in gestational length of several different species that vary in their gestational length. And so for humans, we consider preterm birth as 37 weeks or less of gestation, so birth before 37 weeks. And so that's 92.5% of total gestational length or less that would be considered preterm birth in this graph. And so we can, draw a line that says that anything below this line is considered preterm birth. And you see that for many different species and many different primates, we do see variation consistent with what would be considered, at least in humans, preterm birth. The medical consequences of these individuals being born, these babies of other primates being born prematurely, may differ relative to what we see in humans. but in terms of variation for the trait, it's all there. The only cases where we don't see much variation, and that may have to do with, or is an artifact of the data that we collected, because for some of the species, the data came from laboratory populations where we see really reduced variation, but in some of these cases, we're dealing with specific strains of these organisms rather than examining variation across the entire wild, or rather than examining variation in a wild population. And so what this indicates is that other species could be useful for understanding gestation length and birth timing, and that we don't necessarily have to, or we can get insights into how it is controlled by studying other species. One of the species that we first started looking at was dogs, for the obvious reasons that we had, or people have generated quite a lot of population genetic data. And so we were able to utilize a data set that contained quite a lot of genotypes for that time for many different breeds. And two people in the lab, Sam Smith, then an undergrad, and Julie Phillips, then a postdoc, went into the primary literature and all kinds of breeders' handbooks. They spent a lot of time in the library trying to find breed-specific data for as many different reproductive life history variables as they could. And for control, a lot of these traits are always correlated with body mass. And so we also included data of body mass, which was relatively easy to find. But we collected C-section rates, litter size, stillbirth rate, gestation length for all these different breeds. And then we conducted genome-wide association studies, tried to identify any variants that were associated with any of these traits. And when you do that, you find some major hits. So these are the Manhattan plots for the chromosomes of dogs. And you can see that there are several different loci that have been identified, sometimes more, sometimes less. And what's really interesting is that, so say for C-section, there is a strong hit for a gene called SMOC2. And when we look in the literature, we see that this is a locus that's been associated with brachycephaly. So this is, think of the French bulldog, and it's very short but very large head. Nearly all French bulldogs are born by C-section because the dog's head has been selected to be so big that it can no longer come out through the birth canal. And so what we see is that the variants that seem to be contributing in some of these traits are variants that have been associated with traits that have been selected for domestication. And so that's really exciting from an evolutionary perspective, but perhaps minimizes the usefulness for some of these traits, because the association with C-sections or the association with gestational length may be simply a byproduct or a statistical artifact of association with traits that have been intensively selected through the process of dog breed domestication and selection. So we can also look not just in individual species, but across different mammals and try to see what can we tell about variation in gestational length. It's fair to say that we know a few things about how gestational length evolved, but not much. In terms of its coevolution with other traits, the only trait that's been sort of robustly associated is body mass. All right, so very large organisms have long gestational lengths, very small organisms, shorter gestational lengths, and we don't know much about the genetic networks that underlie this variation. And so we're beginning to do research that aims to find that out. So here is the relationship with body size, body mass. And so if we take the logarithm of gestational length, plot it against the logarithm of body mass, each dot corresponds to a different mammal. What we see is a nice positive allometric relationship that indicates that there is, the larger the mammal, the longer its gestational length. This is something that's well known for some time. What's interesting, at least to us, is the exceptions. So trying to identify lineages or try to identify species or groups of species that seem to vary in that respect. So, organisms that are relatively small but have longer than expected gestational lengths relative to their body mass and so on. And so with that in mind, one lineage that we've begun focusing on are the rodents. The rodents are particularly interesting from the mammalian evolution point of view in that, they make up they're by far the most species-rich family of mammals. They make up 40% of mammals. And then one particular group, the Muroida, that's the group that includes the well-known mice, hamsters, rats, and so on. And they are the most species-rich. Other than the rodents, the other major family are the bats. They're also really interesting from a reproductive life history point of view, but they're perhaps less easy to study than the rodents. So rodents show wide variation in their body size, from really tiny ones to really big ones. And of course, they have a really wide range of life history traits that make them really interesting to study. And so one group that we've begun focusing on and trying to understand are is these three species: the spiny mouse, the house mouse, and the Mongolian gerbil. And the reason is because we believe that the spiny mouse is a really interesting outlier in terms of its reproductive life history. And so when you compare it to the gerbil and the house mouse, you see that in terms of gestational length, it has nearly double the gestational length, even though its weight is not that different. The litter size also differs quite a bit. They usually give birth to 1 or 2 pups, whereas the litter size of the gerbils and the mice are much different. And then the maturity, the developmental status of the neonates is really different. Gerbils and mice, baby mice, they're born hairless, they're largely immotile, they're totally dependent on their moms. The spiny mouse babies, the neonates, seem to be ready to play, much more motile, furred, and so on and so forth. And so we've started collecting the genomes of several different rodents and evaluating their accuracy. Not all genomes that are available in GenBank are that good. And so we started this process of trying to get assemblies that are really good. And in the cases that we don't, try to find tissues so that we can sequence more genomes. So if you have any resources available, please talk to us. We would be interested in sequencing more genomes. And we started constructing genome scale phylogenies of the species for which we have genomes, so that we can do trait reconstruction or try evaluate the coevolution of different traits, such as gestational length and body mass, or others. And then we started analyzing the shared and species-specific gene families. And so we can identify, gene families that are unique to the spiny mouse, or unique to the gerbil, or unique to the house mouse. And what we're finding interesting and potentially promising is that, if you start taking these 300 genes or these 177 genes and do a gene ontology enrichment analysis, you do find that terms associated with pregnancy tend to be enriched in these genes. We don't know how much this will hold up and whether it'll hold up in all cases, but it definitely seems that some of these species-specific genes tend to have functions associated with pregnancy. And so just to summarize what I've told you about, gestational length in humans is a polygenic trait with both maternal and fetal components, and we see some evidence of antagonistic pleiotropy. We infer by studying the genomic regions associated with gestational length and preterm birth that a mosaic of different evolutionary forces have acted on these loci. And so it appears that, at least in terms of selection, reproductive traits are not that different from other complex traits, although we do see different ensembles of complex traits to show different signatures. And then we're beginning to explore rodents as a system for studying the evolution of gestational length across mammals, but this is just at its beginning, and we need more omics and tissue resources to make that happen. And so I would like to thank people that were involved with these studies. I think I pointed out most of them as I was talking along, but not all. So Paul, Ge, Bo, and Lou were pioneers in the genome-wide association study data I talked to you about. The evolutionary signatures of GWASs, this was led by Abby and Abin. With the Abin was in Tony Capra's lab, and Patrick Abbott helped conceptually quite a bit for the first paper as well. The dog work was done by Julie Phillips and Sam Smith, and the rodent work is being led by Theo Danis, graduate student in the lab. We've been lucky enough to be funded by the March of Dimes, as well as through the Barrows Wellcome Fund. and to thank Lou for all his contributions to the field, but also for being the one that recruited us in the field and gave us this really interesting set of questions to study and the means to study them. So thank you so much, and I would be happy to take any questions that you may have. Thanks.
Thank you. That was a great talk. And I invite questions and comments from the audience. In this of this